Thank you very much. صباح الخير جميعا. It's really a pleasure to be among my Egyptian colleagues and friends again. It's always a pleasure to be here in Cairo. So I guess we are going to shift gear completely from the first two presentations, from biomarkers to pathogenesis of MS. And specifically the role of the Epstein-Barr virus, EBV, in MS pathogenesis. And actually this recent research that came out last year, was labeled as one of the top 10 medical scientific discoveries of 2022, the relation between MS and EBV. These are my disclosures. So we already knew that MS had both environmental and genetic etiologies and that they interact with each other. We also knew that almost all MS patients are infected with EBV already. And we also knew that, like all other herpes viruses, EBV can go into a latent phase in the human body, and specifically in the B cells, and can reactivate like other herpes viruses. And it's interesting, this latency within the B cells, especially that we know now from the last 10 years that B-cell depleting therapies are actually very highly effective in the treatment of MS. Now the problem with proving this correlation is that 95% of the general population is already infected with EBV. And to really prove such a causal relation, you're going to need a huge number of patients, millions, and followed for decades. So actually, this human experiment was actually found by the group in Harvard, Alberto Ashiro and his group, and that experiment of nature was actually the US Army. In the US Army, of course, you have young people enrolled, and they take blood samples from those persons when they come into the Army, and then every single 2 years for decades. So here you have a young population which is followed over decades with blood samples available every 2 years. So what the Harvard group did, they went in and they looked at those army recruits and saw how many of them developed MS. And over those decades, 955 of them actually developed MS. They went ahead and looked at their blood sample, at least 3 blood samples before they developed MS, and later on. They found a total of 801 MS patients that fulfilled the criteria, that they developed MS later on and already had 3 blood samples available from before. So they went ahead and compared them to 1,566 controls from the same army, of course, age match and so on, that did not develop MS. And the first important finding was that out of the 801 MS patients, as expected, all of them were EBV positive except for a single patient. But the most important part of the study is what I put here at the bottom line, that 35 people that developed MS later were actually, when they entered the army, were EBV negative. And they took those and compared them to 107 controls that did not develop MS later on, and who were EBV negative at entry. And these are the main results of the study. If we look here, the patients that were EBV negative and developed MS in green, almost 97% of them got infected before developing MS. So their blood sample turned out from negative to positive antibodies before developing MS. While those that did not develop MS, only 57% of them seroconverted. Now, to make sure that this is not really a haphazard or that the period preceding the onset of MS has immune dysregulation that can lead to increased viral infections, they looked for CMV. And sure enough, such a correlation was not found with CMV. Whether you develop MS or not, the seroconversion to CMV is the same. So taking this number, it seems that, number one, if you are not infected with EBV, your risk of developing MS is almost nil. While if you get infected with EBV, your risk of developing MS increases by almost 32 times. And remember that up till today, the highest risk factor for MS was the HLA-DRB15, and that risk increased the risk of MS by threefold. So now we have a risk factor increasing the risk of MS by almost 32 times. They took the study further by looking at the neurofilament light chain, which Professor Fox just talked about. And as we know, neurofilament light chain reflect the degeneration of the axon within the central nervous system. And usually it was found that they precede the clinical onset of MS by almost 7 years. So the degenerative process starts years before the first symptom. And when they looked at these neurofilament light chain, they found that the rise in neurofilament light chain, which is the start of the degenerative process in MS, always followed the EBV infection. So the EBV seroconversion always preceded the onset of the degenerative process. Another strong correlation or indication about the causative role of EBV because it's preceding the disease. And of course, such a correlation was not present in patients that did not develop MS. So the main question regarding this research was, are we dealing with an actual causality relation, or is it a haphazard, basically, correlation? And the issue of reverse causation. So beside the CMC seroconversion issue, besides showing that the serum neurofilament light chain and the degenerative process follows and does not precede the EBV infection, they looked at all possible viruses that can infect the humans. And they looked at 30 MS patients and 30 control and checked for all possible viruses. And again, they found that the only virus correlating with MS was EBV, a seroconversion of EBV, and none of the other viruses. So the conclusion of this very, very important paper is that, number one, EBV is likely the leading cause of MS because of all the points I just mentioned. And actually, 𝐬𝐩𝐸 says in his paper, ultimately in the discussion, that we thought that MS was an autoimmune disease of unknown etiology. Now we should start thinking of MS as a complication of infection with the EB virus. And I want to remind you that back in the 60s, before we discovered that cigarette smoking is the main cause of lung cancer, the first study to show such a correlation found that cigarette smoking increases the risk of lung cancer by 25-fold. And now here we have a study showing that EBV infection increases the risk of MS by 32-fold. So of course, it seems that EBV is a necessary condition for MS. There is no MS without a previous EBV infection, basically. But, of course, not everyone that is infected with EBV will develop MS, because 90–95% of the general population is infected with EBV and do not have MS. So, EBV has to interact with many, many other factors. Basically, the sex, more likely to be in female. In older age at EBV infection has also been shown to carry a higher risk of MS as opposed to a very early infection during childhood. And of course, many, many other risk factors that we know for MS, including obesity, cigarette smoking, stress, and so on. So it's a multifactorial etiology, but we have a necessary condition, which is the EBV infection interacting with other things. Genetics are also important. And we remember that the HLA-DR15 gene was basically the most important risk factor for MS. And interestingly enough, studies have shown that this HLA type will predispose to dysfunctional T cells, which will not be able to regulate or to destroy the EBV-infected B cells. And this is why an infection with the EBV in a patient that already has the HLA-DR15 increases the risk of MS 7 times as opposed to patients with other HLA typing. And this is really one of the nicest examples about how environment and genes can interact in producing a disease. So the question is, how can EBV cause MS? There are many theories, but the one that's probably the most accepted is the molecular mimicry theory. And the molecular mimicry theory, basically, if you look here, this is just a cartoon, that there are certain viruses, let's say with this green antigen on the surface, that their surface antigen look like antigen within the cells of the human body. And once the immune system recognizes this antigen on the virus, they trigger usually the whole cascade of T cells, B cells, and so on, an antigen-presenting cell, leading ultimately to the destruction of the cell infected with the virus. However, once we have antigen within the body that look like these antigen on the virus, this immune system that is triggered by the virus will go in, destroy the virus, but at the same time destroy the cells that contain this antigen looking like the viral antigen. So putting it simply, the immune system basically gets confused between this antigen on the virus and the antigen on myelin or wherever in the central nervous system, and this leads to a double attack and destruction of the myelin or whatever antigen in the central nervous system. So this is putting it in a cartoon way. Actually, the original Harvard paper was published in January. The following ones in February, another paper came up from Stanford looking at this antigen in the central nervous system that could be the central of this molecular mimicry. So what they did is they identified an antibody in the CSF of MS patients, MS39. They isolated it and found that this MS39 antibody binds strongly to the EBV, to the EB virus, and specifically to the EBNA1 protein on the surface of the virus. So they went ahead and probed these antibodies against almost 16,000 human proteins, spanning more than 80% of the proteins in our body, and they found specifically one protein, 𝐬𝐩𝐸𝐿-CAM, that actually bound to this antibody. And this 𝐬𝐸𝐸-CAM molecule was present on astrocytes and oligodendrocytes. So here we are discovering a CSF antibody in patients with MS that can bind to the EB virus and to a central nervous system protein present on the astrocyte and oligodendrocyte, which is basically what we were looking for in the, to confirm the theory of molecular mimicry. And when they immunized the mouse model of EAE with EBNA1, the protein on the EB virus, sure enough, it led to the production of anti-EBNA1 antibodies and anti-glial-CAM antibodies, producing a more severe EAE disease in these mice. They went back to the human source and they checked the CSF of MS patients and found that 20 to 25% of them actually have the MS39 antibody. So did we finally discover this elusive antigen, which is the cause of MS in the central nervous system? I think it's still too early to say, but I think that we're going to need more and more studies to confirm these findings. So how would this new theory affect our approach to treatment of MS? So what can we do if we know that? Number one, we can have a vaccine, right? We can vaccinate all children against EBV, eradicate EBV, and probably eradicate MS, and that would be the cure, just like we eradicated poliomyelitis or smallpox. Of course, such a study is not going to be easy to carry. We're going to need a huge number of patients with a very long follow-up to confirm these outcomes. The second point, we can use antiviral drugs and try to target these EBV-infected B cells. Now remember that like all other herpes viruses, like herpes labialis, for example, it goes latent and then it reactivates, or herpes zoster radiculitis. So maybe that the EB virus is latent in those B cells, and every time it reactivates, it's actually a relapse. And once it compartmentalized within the CNS and produces a chronic activation, then this is progressive MS. So in that case, using antiviral drugs might actually be a treatment for those relapses and those progression, of course, assuming we can have an antiviral drug that can cross the blood-brain barrier in progressive MS. Such an antiviral drug is not available yet against EBV. Tenofovir might be a best example, but I think there is ongoing research now to try to design an antiviral drug against EBV. And finally, and this is where things are really developing the quickest, developing what we call EBV-specific T cells, which means T cells that are programmed in vitro to destroy EBV-infected B cells. The initial trial for a vaccine are already ongoing. So Moderna, using the same mRNA technique that they use for the COVID-19 vaccine, have already launched their trial of EBV vaccine, aiming to recruit around 270 persons. The NIH is already launching another EBV vaccine trial, this time targeting the GP350 protein, which is on the surface of the virus. So we're going to have to wait and see for these results, the safety of such vaccine, the efficacy of such vaccine before going into these large vaccine trials, which will have to be followed for a very long time. Treatment of MS, again, the trials have already started based on the EBV theory. And initially, as I mentioned, the easiest way was to use these EBV-specific T cells that can destroy the EBV-infected B cells. And such a trial using autologous CD8 cytotoxic T cell, autologous meaning they took the actual T cell from the body, programmed them against the B cell that are infected with EBV, and then reinfuse them intravenously into those patients. The initial trial was for 10 patients with progressive MS, and sure enough, 7 of the 10 patients actually showed improvement, and 6 of them had actually objective improvement, as we can see here on the fatigue scale. And in 3 of them, there was reduced intrathecal IgG production. There was no significant adverse event. A second trial used the ATA188, and this is really a different approach using allogenic T-cell immunotherapy, which is manufactured through multiple donors, not manufactured, but collected from multiple donors. It is allogenic, so it's not autologous from the patient, and these are programmed, T cells are programmed to destroy again EBV-infected B cells. An initial trial was done in progressive MS again. Most of these trials are being done in progressive MS so far. In 24 patients, they got the infusion of the ATA188 for 12 months, and then some of them were followed for a longer period, up to 39 months. And out of the 18 patients followed for a long enough period, 9 actually achieved sustained disability improvement. And when they look at the MTR measures of some of these demyelinating lesions, there was evidence of remyelination. A phase 2 trial now, it's not showing here, of ATA188 called EMBOLD is being launched now as a phase 2 trial treatment of these allogenic T-cell EBV infected B cell specific as a trial in progressive MS. So I want to remind you here about the history of gastric ulcer and Helicobacter pylori. If you remember, in the 80s, we used to think that gastric ulcer is due to hypersecretion of acids. And we used to give the patient a high dose of antacid, PPI, and even sometimes do gastrectomies for severe bleeding. And then came Moran and Marshall, and they found out that actually there is a bacteria called Helicobacter pylori in the stomach that actually is inducing this hyperacidity, and a simple antibiotic cured gastric ulcer, and we don't ever do anymore any gastrectomy. And this, of course, was awarded the Nobel Prize in Medicine in 2005. So the question now, is MS also a complication of a viral infection that we might ultimately lead or eradicate by treating the EB virus? I guess the next few years will tell us. Thank you very much. And of course, you're all invited to our next Menactrums Congress that will be in Abu Dhabi in December 8th.